The last topic that I want to cover in our section on phylogenetics is a really important one for the topic of Evo-Devo, and it requires a review of, or at least making sure you know and understand this concept of homology. So remember, homology is similarity of characteristics due to inheritance from a common ancestor, right? So pretty simple concept, right? The wing of a eagle, the wing of a parrot are similar because they share a common ancestor with a wing. The wing of a bat and the wing of an eagle are different in many ways, right? Because as a wing at least, they come from a different common ancestor. And so we've established a little bit more of the fine details of homology, and we've made some extensions of those. So we talked about serial homology, right? And this is looking at serial homology at the genetic level. Okay, so it's important that we understand this. So I've got a diagram up on the screen here. In this diagram, the black lines, kind of the big fat phylogeny, represents the species history. Okay, so we've got species A, B, and C, and you can look at this very easily and see that species B and C are closely related to one another, and they share a common ancestor right there. And then species A is related to those two as a group, and they share a common ancestor right here. Now, in addition to the species history, I've also shown internally inside the species history, I've shown the gene history. And I've done this, I've color-coded it, so we've got red lines representing one branch of this gene family. I've got green lines showing a second branch. And we've got another node, an additional node that doesn't represent a speciation event. This represents a gene duplication event. So I've labeled that as a gene duplication event. Now, all of these genes are related to one another. They all are going to be similar to one extent or another, and they all can trace their history back to that one ancestral gene here with the green line that we've shown here. Okay, so they're all homologous. However, there are some important distinctions at the genetic level about homology. Now, all genes that come from a common gene ancestor that we identify are part of the same gene family. So genes 1 through 6 would all be part of the same gene family. And you already know about some gene families. For instance, the globin superfamily of genes. And within that, there's a subfamily called hemoglobin. And then if you know something about hemoglobin, you know that there are different subunits that make up the final functional hemoglobin protein. So there's alpha hemoglobin and beta hemoglobin. And the names give us some indication about the history of those genes. And so let's look at the different possible histories. So notice that we have an ancient gene duplication event here, which means that the ancestors of these three species carried 2 copies of this gene. Now that is a form of serial homology at the genetic level. We have a duplication event, and we end up with 2 genes, and initially they're going to be right next to each other on the chromosome. Later they might get shuffled and moved around. But initially those genes are going to be identical. So just like we talked about duplication of a structure like a leg in an arthropod, we're going to end up with redundancy. Initially those 2 identical copies of the gene are going to be doing the exact same thing. Later, they may diversify. New mutations may occur in one of those genes and not in the other, or in both. And we get diversification of function of those genes. Okay, but the end result is that species A carries 2 copies of this gene, species B carries 2 copies, and species C carries 2 copies. And I've just numbered these, right? The numbers don't have any indication of how they're related to one another. They're just for our convenience sake, and we'll be looking at these. Now, after gene duplication occurs, a number of other things can also occur. One of which is we could end up with loss of one of the copies of the gene. Now, when I say gene loss, that can take a number of different forms. Gene loss could mean that a piece of DNA due to a mutation is just absent from that species. So species C still carries 1 copy of this gene. That would be part of the red subfamily, but it no longer carries this second family if it lost that chunk of its chromosome. Okay, now remember, all of these mutations are like any other mutation. They can be good, they can be bad, they can be kept, they can be maintained. So all of your good, what you learned about mutations in evolution still applies here. But if it was lost and it was a beneficial thing or was kept for one reason or another, that loss was or that part of the chromosome was no longer there, then we'd end up with fewer members of this gene family in species C. And that would be an important part of their history. Okay? So another way that we could have what we call gene loss is if that sequence of DNA was still there, but it no longer functions as a gene. So it's still part of the genome, and maybe if we sequence the whole genome and look for sequence similarities, we could still recognize it as part of that gene family, but no longer a functional gene. And that would also be technically a gene loss. So realize gene loss could mean loss of the entire sequence of DNA or just a mutation, so that piece of DNA no longer functions as an expressed unit, and it would no longer therefore be classified as a gene. So there is specialized terminology to distinguish between these different types of homology that we see in gene families. Okay? The first term, the most common form of homology, is called orthology. Ortho means straight. If you want to think of orthodontist, and that's helpful, great. But orthology means that two genes are similar to one another because they differentiated at a speciation event. That's the easiest way to think about it. So gene 1 and gene 3 are homologous to one another because we get back to their common history and they broke up when the species broke up. Okay, so alpha hemoglobin in humans and alpha hemoglobin in chimpanzees are orthologous copies of the same gene. Now, paralogous copies means that they are similar to one another, they share a common ancestor. However, they do not unite back into a common ancestor until we get back to a gene duplication event. The easiest example of paralogous genes is if they are found in the same species, they're almost always going to be paralogous. Now notice if I trace the history going down the green line and the red line, that those two lines don't connect back together until we get here at the gene duplication event. And so because 1 and 2 come from the same species, you would automatically know that they're going to be paralogous. But if you really wanted to see it, you could figure out the gene history and the species history and then trace back their common ancestor. If it's a duplication event, they're paralogous. If it's a speciation event where they break up, then they are orthologous. So notice if I look at 5 and 1, I get back all the way again to the same gene duplication event, and therefore they would be paralogous. Now, there's a subcategory I'm just going to address. You need to know the definition, but we're not really going to be dealing with it in evo-devo. It's not an important pattern or important phenomenon when we're looking at evolution and development of animal form. And this is xenology. Xenos, the prefix here is the Greek root word for alien or foreign. And xenology occurs when we have a horizontal gene transfer. And so if a gene traces its history through a horizontal gene transfer event, it's still going to be similar to this family of genes, but now we refer to it as xenologous. And xenology is a little bit more important than we originally thought, and there are some interesting studies recently that show higher levels of xenology than we thought in animals, but still doesn't play a really important role in evolution and development. It is really, really important in bacteria. Lots of bacteria have high levels of horizontal gene transfer, and so we get high levels of similarity due to xenology. Okay, but again, for our purposes, as long as you know the definition that xenology traces history of genes, it's a form of homology that has a horizontal gene transfer connection. That's all you need to know for our purposes. Okay, but in developmental genes, remember we talked about those 3 classes of genes. We talked about housekeeping genes, we talked about tissue-specific genes, and then the small subclass of genes that we're going to look at more and more in our coming lectures that we call toolkit genes. These are the genes that form the blueprint, the ground plan for building an animal. And in many of these genes, they have gone through many, many duplication events. So notice on this diagram, I've shown a single gene duplication event. But in many cases, there are many, many, many gene duplication events that result in very large families of genes. And developmental genes, these toolkit genes, are one of them that have had multiple duplication events. And we'll talk about some of those important duplication events and where they occurred in the history of animals in coming lectures. But just as an example, I've put together an alignment of 8 different genes that are all part of the Hox gene family. And we're going to focus and talk a lot about Hox genes in the coming weeks. So you don't need to know the names for now. I'll probably have you learn the names in a coming lecture, but Notice that we have 8 different genes that have a pretty high level of similarity. You can see the patterns and shared similarities at the DNA level in this region of the genome. This, by the way, is called the homeodomain of these genes. Notice they're all from the same species. So even though they're similar, and even more similar, this is a protein alignment of this same region, but just translated to proteins. They're even more similar at the protein level. And so even though they're all similar, they all come from the exact same species. These are all Drosophila melanogaster. And so their similarity can be traced back to an ancestral gene, but not until we get much more anciently into a gene duplication event. And so these are all part of the same gene family, all are the result of multiple gene duplications, resulting in 8, and they're actually more than this, but these are the 8 important ones that are still kind of classified as standard Hox genes. But there are all these duplication events leading to these many members of the same gene family. Okay, so to kind of summarize, review, give you a little bit more vocabulary, I'm going to show you this image. I've provided it in the PowerPoint if you want to take notes on it or highlight it, but I'm going to look at some things and make some changes to it, and I'm going to do this in Illustrator. It just makes it a little easier. So here is this same diagram in Adobe Illustrator. Okay, and it's basically the same one you saw before with a single gene duplication event anciently down here, and then one of the genes being lost. Okay, now notice that if I just look at the gene history, I'm going to have these 2 phylogenies, internal phylogenies, that I could look at it as a single phylogeny showing the history of the genes. And this is going to be a mix of orthologous and paralogous relationships. Okay, we talked about orthology, we talked about paralogy. The nouns for those, if 2 genes are similar due to a gene duplication event, we would call those 2 paralogs of one another. If they're similar because they broke up at a speciation event, we'd call those orthologs. So same basic terminology, just whether it's the adjective form or the verb form. Now notice, and I'm going to color code these, that actually these are mislabeled. Let's change this. This should be 1, 4, and 5. Sorry for that mistake. I'll change that on the PowerPoint also. It should be 1, 3, and 5. The way I've numbered them, it ends up that all of the odd numbered genes are more closely related to one another. And then this would be 2, 4, and then the pseudogene, which is a good vocabulary word. A pseudogene is a gene that's been lost because it's no longer expressed. It's still there in the genome and we can identify it, but it's no longer an active gene. So we call it a pseudo again, pseudo meaning false or fake gene. Okay. Now notice, if this doesn't make sense to you how I get this diagram from this, I'm going to color code it. So notice 1/2 of the gene family, the green side, 1, 3, and 6 here. 1, 3, and 6. Oh, I did this weird thing where I made the green go on one side. So it's actually one last correction here. 1, 3, and 6, 2, 4, and 5. Okay, now I've got everything arranged the way it is. So let's color code it. So notice 1, 3, and 6 all share a common gene ancestor right there. They're all orthologous to one another. And then they connect into the other part of the gene family right down here at the gene history. So I could color code all of these lines green. Let me do that again. Got to make sure I'm on the right setting here. There we go. So that represents the green gene history here. And again, we can maybe put a little X somewhere in the line here if we wanted to represent that there was a pseudogene, that we'd lost that one at some point. But if we could still identify and sequence it, we could still put it in an alignment and compare. And then, of course, this other half of the genome we would recognize as being the red part of the history. Okay? And so on this gene phylogeny, some of the nodes represent speciation events. I'm just going to put little circles over all of the ones that represent a speciation event. And let's just make them black. That's probably the easiest thing to do. Okay, so we've got a node there and there. And those 2 nodes correspond to this speciation event. That's where 3 breaks up from 6, 4 breaks up from 5. So there they are on our gene history. And then we also have these deeper nodes there and there that also represent a speciation event. They correspond to this deeper node where 1 breaks off from 3 and 6, and 2 breaks off from 4 and 5. And then finally, at the very end, I'm going to change shape because this is a different event. This node here, although on this phylogeny it looks just like the other nodes, it corresponds to that gene duplication event. And so when we see a gene history, we might have both speciation events and gene duplication events represented in that gene history. And if we didn't know the history of the species, if we knew what species these came from, but we didn't have a phylogeny for those species, it would be maybe difficult to determine. And this becomes especially difficult to determine if there are many duplication events or duplication events and multiple gene losses. It can be a very difficult history to infer, and it's a very complex process that we won't really get into much in this class. We'll talk a little bit about it in the labs, but the process itself we won't worry about for the purposes of this lab. Okay. Now, the last term here that I have, just to remind me of our details, the things that we need to talk about before we finish up, is neo-functionalization. Now, it's a long word, but the root words are pretty self-explanatory. Neo-functionalization refers to a new function for a gene. So when we have a gene family with separate branches, and one of those branches evolves and takes on a new function that wasn't there in the ancestor, that's called neo-functionalization. And this is a genetic connection to something that we've already talked about. So when a gene duplicates, you can think of those as like serial homologs. Now, the technical term is paralogs, and initially they're identical, and they're right next to each other on the genome. But then if one of them takes on new function, we could still maintain the ancestral function in one copy, and the now duplicated version could take on a new function. And so this is the most important mechanism for the evolution of new function basically over the entire genome, but especially important in developmental genes. And it mimics what we talked about at the morphological level, right? So the initial advantage of a gene duplication event, just like the duplication of a structure, is simple redundancy. We've got 2 copies, and that can be good in some cases, right? But the added long-term advantage is now that we have multiple copies, we can change one of them, right, through mutation and natural selection, genetic drift, and all those great things you learned about in evolution. We could change the function of one of the copies, but still maintain that ancient ancestral function. So we get diversity without losing the original, probably very important function, especially for developmental genes, without losing that very important function. And so neo-functionalization of duplicated genes is a really, really critical outcome and an important trend that we see across these developmental toolkit genes.